I think with that, I think we'll get started. So, hi everyone, and welcome to the TIA Centre seminar. My name's Adam, and I'm a postdoctoral research fellow here at the TIA Centre at Warwick. For those that are new to our seminars, in these seminars we aim to invite researchers from across the globe to present new and exciting work. And to get started today, I'd like to introduce Dr. Korsuk, and now I apologise if I don't pronounce your name properly here, but Sirinukunwattana. You might want to correct me there.
No worries. I think.
So Korsuk is a postdoctoral research assistant at the Institute of Biomedical Engineering, in the Department of Engineering Science and the Big Data Institute at Oxford University. His main interest is the applications of image analysis and machine learning in medical research. He has been actively involved in several research projects in computational pathology, focusing on morphomolecular correlation and cohort indexing in colon, breast, and prostate cancers. He completed his doctoral research in the group in the Rajput at the University of Warwick. The title of Korsuk's talk is Image-Based Consensus Molecular Subtypes of Colorectal Cancer. So Korsuk, if you're ready, feel free to share your slides. I might need to just quickly make you a host. Yep, there we are. You should be able to share your slides now. And yeah, feel free to begin.
Can you see the slide okay?
It's still loading at the moment.
Is it coming up?
Yeah, we've still got a screen that's just saying loading. I don't know if it's...
Let me try to reshare it again.
Thank you.
Not coming up?
No, we've still got a blank screen here.
Oh dear.
Do you want to stop sharing and see if you can reshare again?
Okay, let's try again.
Third time lucky. Yeah.
Okay.
There we are. I can see your slides at the moment.
Perfect. What about this?
And now they've gone.
Oh, but here's okay.
Yeah, maybe if you just reduce the size of the margin on the left. Yeah.
Yes. So basically, how can I close this?
Or maybe there we are. Perfect.
If I do it like this, can you still see the screen?
Yeah, we can see that. Okay.
Okay. So thanks for the invitation. So I'm going to talk about this research work, which is published for quite some time now. I think it's 2021. And it's a joint work between myself, Enrique Domingo, Tim Mond, Jens, Richard, Victor, and the rest of ESCORT consortium. And it's about image-based consensus molecular subtype of colorectal cancer. So let's start from looking at the statistic of colorectal cancer. So I said that it's the second most common and the third most common type of cancer-related incident in the world, in male and female. And there is about 1.4 million new cases every year, 700,000 deaths annually as well, and mostly in population of 40 plus, with the survival of about 57%. I think this is the survival statistic is from the UK, which is slightly higher than other places in the world, I think. Then, let's go to the next one. So what is the current standard of diagnosis? So what is used for staging right now is the TNM staging. T stand for primary tumor, N that is the number of lymph nodes, and M is metastasis, whether the disease has spread to other part of the body or not. So this is an anatomical system. For instance, in stage 1, what you have is localized disease. I'm not quite sure if you can see my pointer or not. No, I guess not, but let's do it like this. So you have localized disease. Tumor is still localized on the inner part of the colon wall, no spreading. The most logical treatment option would be surgery. And then in stage 2, you can see that the tumor has sort of penetrated through the colon wall. So localized advanced disease still. So you would have surgery and then followed by chemotherapy. And stage 3, apart from localized advanced disease, there is also involvement of lymph nodes as well. So the same kind of treatment, surgery plus adjuvant chemotherapy. And then in stage 4, so you also have sort of metastasis. This is already spread to other part. So it's going to be quite sort of advanced combination of treatment, surgery, chemotherapy, and perhaps targeted therapy like immune therapy if you have the genetic profile that would be benefit from the immune therapy. So that's the TNM staging in a nutshell. But the problem with this anatomical system is that it's not reliable to use for prognosis and treatment decision. For instance, when you look at the subpopulation in stage 3 versus subpopulation in stage 2, you will find that some of the subgroup in stage 3 actually doing better than those in stage 2. And also it doesn't really tell you what option of treatments a patient should get. So it doesn't really tell you what benefits of the treatment basically specific to each patient. So that's the problem. So now, excuse me. Now let's look a bit at the biomarkers that has been published so far in literature. So these are the list of biomarkers from the latest WHO Blue Book. It lists quite a few biomarkers over there. But if you sort of put these biomarkers into 3 main categories, so you would have diagnostic markers, predictive markers, and prognostic markers. Diagnostic markers basically is used to ascertain whether patients have disease or not, and in this case, whether patient has colon cancer or not. Very simple. Predictive markers, this is the marker that you want to use to select treatment options to a patient, because it would tell whether a patient would benefit from that particular treatment or not. And then for prognostic markers, that's determined the outcomes after treatment, whether the patient would survive long enough, or would they have relapse of disease later on or not. So there are quite a few markers over here, but the current one that's used in our sort of the NICE guideline using NHS standard routine sort of treatments. So that would be MSI, BRAF, and KRAS. So only 3. The rest are not yet recommended. So now the problem of trying to introduce these sort of new markers into the standard care is that they are all expensive in order to introduce this gene panel, new gene panel, you need to have to set up a new sort of workflow, and that's quite expensive to do. And of course, the turnaround time would be more than 48 hours. So that's quite significant. So we want to have something that is cheap, so quicker. And what would be the alternative solution over here? Of course, you have histology slide, which is produced in routine workup. Every patient would have this slide. So the idea over here would be to use histology, the morphological information in histology slide as a surrogate to predict molecular profiles. And if that is possible, then you can think of the way to introduce the sort of image test first. Like the first layer, you use image test because it's fast, cheap, but then it might work on a proportion of patients. There would be some that could not be ascertained with confidence using image tests, right? Then those patients, then you can think of other tests down the line. So image and then morphology, sorry, the image and then molecular tests. So that's the idea. Any questions so far, by the way? I've been, no.
I don't think there's any questions so far. Thank you very much.
Okay, yes. If there is any question, please feel free to interrupt. So this is the work that came out more or less at the same time from Jacob Kater. Very nice work that predict MSI microsatellite instability from histology slide. And that showed that it is possible to correlate morphology with molecular or genetic profile. And yeah, so let's talk about the consensus molecular subtype. So this CMS has been proposed by Guinea in 2015. Is based on gene expression data clustering. And the reason that we pick CMS as compared to other like genetic or molecular signatures is because CMS provide a very well biological interpretation of different sort of like pathway involved in the cancer biology. As you can see over here, there are CMS 1, 2, 3, and 4. And for instance, in CMS 1, you have immune high activities. CMS 2 and 3, they are mostly epithelial. Of course, there is like Win-MYC activation in CMS 2, KRAS mutation in CMS 3. And CMS 4, that's mostly stromal activity, TGF-beta activation, and so on. In terms of the worst survival outcome, you would have CMS 4, and then CMS 1, that's the worst survival after relapse. Generally, CMS 1 has very good outcome, but if the disease relapse is the other way around. So that's quite interesting. So the other thing to note over here is that if you sum the numbers on the top together, that would equate 87%. So that means that another 13% cannot be classified into any of this bucket. So we would call those like molecularly unclassified samples. So let's start with the work. I think those are all the introduction. So in the work, we want to predict CMS label for each, well, given a whole slide image. So we have 3 cohorts over here: Focus, TCGA, and Grampian. Focus and TCGA, they are full resections, as you can see from the example images over there. And Grampian, that's primarily biopsy. So in terms of the tissue surface, Grampian would be much smaller. And you see over here in the above panel that in terms of the data distribution, CMS2 and CMS4 would be the majority. And then 1 and 3 would be the minority classes. And of course, you would have unclassified samples in gray as well. So we use Focus to train our algorithm and use TCGA and Grampian to validate. And the reason that we use Focus is because we have this very well aligned, spatially aligned between the image and the location where we take the DNA and RNA readout from. So, I mean, it's quite a well studied. So given a tissue block, first we cut one slide, imaging H&E, and then 4 slides down that's used for RNA extraction. And then we cut another slide, image it, H&E, and then 8 consecutive slides down that's for DNA. So we know exactly, the location of the DNA and RNA extraction. So that's a good starting point. In terms of the algorithm training, what you have as the input data is this whole slide image. There is a pen mark to demarcate the region where DNA RNA extraction is performed. Of course, we have only one label per slide. So we have only one CMS label per slide. But we know from previous research work that if you sample a small region of tumor and make CMS call, you would get, and then compared to taking another read from the region from the same tumor, but perhaps a little bit further away, you would get another CMS call. That means that there is heterogeneity of CMS within the same tumor, and therefore it makes sense to hypothesize that different sort of like image tile would have different CMS label. So what we do is basically extract a lot of tiles out of the mark region, and then we train a simple, I think, I forget which one I used for this classification. Is it? I think it's Google V3. So for each tile that we extract, the label that we use would be the label of the slide that those tiles are extracted from, and we use only Focus to train the label classification. But at the same time, we know that these cohorts are quite heterogeneous. So what we want is to be able to have our classifier learn features that are cohorts diagnostic so that it can generalize well. So we also use tiles from TCGA and Grampian to do domain adversarial training as well. I will go into the detail. We trained 5 different sort of models to perform ensemble. So it's like 5 different experts voting basically. So in terms of the domain adversarial training, this is based on done in 2015. If you look at the modern deep learning architecture, so you would have 3 parts, right? Input data, and then the network, which mostly feature extractor. And then you have at the end sort of like fully connected layers that perform classification. So we have 2 streams of data over here. The one in blue, so that's the tiles from focus cohorts that we use to train our CMS classifier. And the other stream is for domain adversarial training. We feed in focus, Grampian, and TCGA without CMS label. But the task for the classifier is to predict which cohort those data are from. And over here, what we want is to penalize the classifier to not be able to tell what features they are, like what data or what cohorts they are coming from, as a way to encourage the feature extractor to learn features that are cohort diagnostic. Any questions so far, by the way? Sorry, I think you are muted.
So we've got a quick question from Sarah in the group chat. She said, how did you assess that your classification was not cohort diagnostic before integrating domain adversarial training? Is it a lack of generalization on one external test set?
Sorry, can you repeat again?
Yeah, of course. So I think she wanted, what she's asking is, how did you check that your network wasn't, it wasn't generalizing very well, basically, that you needed the domain adversarial training?
Oh, yeah, I'll talk about that. So in next slide, I will tell you how I checked that. So in the next 2 slides, I think.
Okay, brilliant. And I suppose I've got a quick question here. So you've how do you translate from this training where you're trying to predict poorly which data set it's from to actually training for the actual task?
So
in this network, you're putting in 2 images, right? And you're saying which data set it's from.
Yes.
And you want it to perform badly.
You want the classifier that predict the data set to perform poorly.
Yeah.
Why?
So how do you then go on? Is this like a first step of the? So then how do you go to then train to predict the actual outcome?
Yeah. So you train 2 classifiers simultaneously.
Okay, got you.
Yeah. Sorry, I didn't make it clear. So for Sarah, I think.
Sorry, I also have another question on this slide.
Sure. Yes. Sorry.
So with focus data set, you have the tumor regions annotated. Obviously, you extracted patterns from the tumor region only, if I'm not wrong, right?
Sorry, it's difficult to hear if you.
Oh, can you? All right. Can you hear me right now?
Okay, better. Sorry.
Yes. So my question is, for focus data set, you have the tumor regions annotated, right? Yes. And you extracted patches only from tumor region. Whereas, as far as I know, for TCGA, well, you don't have that out of the box. So did you also do that for the TCGA dataset manually, or did you use all the regions from TCGA? Because in that case, the adversarial learning might be differentiating between tumor-based features and, say, normal tissue features or stroma features. You know what I mean? And also, this also goes for the inference as well. Did you do inference only on tumor region on TCGA, or how did it work?
That's a good point. I forgot to mention that for TCGA, we have annual annotation from the same pathologist that do the annotation on focus. It's a spin central review. So everything is consistent across different cohorts.
Oh, that's
so,
great.
Yeah.
Okay.
Yeah.
Thanks.
Yeah.
Brilliant. Thank you.
No worries. So I'll just talk about, I mean, I think this is something like everyone is it's known quite well, I guess. Like if you have two data set, over here it's just a toy example that you have MNIST, the classic MNIST on the left, black and white, 0 to 9, while on the right that's MNIST M, so it's quite colorful as compared to the black and white. And if you train feature extractor and then you visualize, so basically project the features at the top level onto 2 dimension, what you would get, I think, is the red, that's MNIST, and in blue that's MNIST M. So you see that the features are quite separated. While if you use the domain adversarial, then the feature would mix better together. So in a way, that's cancel out any sort of like cohort specific feature. And the way that the way that I used to check this priority is to do very simple clustering analysis. So initially, without domain adversarial training, if you do clustering analysis and you look at the consistency of of features across different cohorts, you will see that they are not quite consistent. But after domain adversarial training, you can see that the enrichment of ties that belong to, say, CMS1, you would see that most likely is in cluster 1 across 3 different cohorts. Similarly, in CMS2, ties that belong to CMS2 will be enriched in cluster 3, 2, 3, 4, 5, and so on. So in this way, it's sort of like visual checking, perhaps, that the features that you get is more consistent across different cohorts. Hope that answered the question of Sarah. Okay. So now for the inference step. Given a new unseen data, again, extract tiles from the annotated regions. Each tile is passed through 5 different models. And of course, each model might predict the label of the tile differently. And you sort of like put all the tiles together. And for instance, over here, you would be, over here it's predict that CMS1 is about 65%, and CMS4 about 30% from this slide. Any question, by the way? Sorry.
No, I think we're okay at the moment. So yeah, please feel free to carry on. Thank you.
This is the classification in terms of the AUC for different CMS classes across 3 cohorts. So the average AUC is roughly about 0.85%. So it performed, I think, consistently well. It can be better. We have higher data set. I mean, we have more data set now, and I think you would see that once you have more data, then it would perform generally better. I think the part that is more
Sorry,
exciting.
Korsuk,
Yes.
do you mind if we ask a couple of questions?
Yes.
I
Yeah,
think most of us got
sorry
one.
again, yeah, thank you.
No.
So, but when you were explaining data, there was, so you had 4 categories and 1 unclassified ones.
Yes.
So I wonder if you've used them in the training at all, or you just neglected them?
We neglected them. We didn't use those unclassified.
All right, okay then, thank you.
Yeah, it's fully so it's like fully supervised settings.
Full class classification.
Yes, full class classification.
Thank you.
We've also got a question from Alex in the chat. Is the area of the curve slide based, or is it patient level?
It is, sorry?
Is the area of the curve slide based, or is it patient level? Is the question in the chat we've got.
It is a patient level.
Okay. We've also got another follow-up question, I think, from Sarah. She says, yes, thanks. Just a follow-up question. Was there any visual evidence then that one cohort was substantially different than another? For instance, morphology or staining, like the examples you showed with the MNIST datasets.
Yeah, so the TCGA, of course, is scanned by I think apparel mainly, and well, there's quite a few problems with TCGA, I guess. Let's start from focus. So focus, we have data from about 100 different. So the data is sourced from about 100 different institutes across the UK. But they are prepared and staying within the same labs. They are prepared in different batches. You would generally see batch effect. However, this is the characteristics, as you can see. I think I can go back quickly. That there would be sort of like bluish over here as compared to TCGA, which is again prepared totally in different lab. And it's scanned by apparel over here. So the file format, the stain characteristics, everything would be different. And when you compare to Grampian, Grampian has quite a lot of artifact because this is biopsy. So you can already see the heterogeneity of the data over here. If you were, I guess, like when we try to do it on focus alone, it's sort of like trying to generalize to other cohort. You would see that they are performing all right, but it can be performed, like you can see that you need to correct for those batch effects somehow. And that's why domain adversarial is quite useful here.
Well, thank you very much.
Yeah. So let's go back to here. Because to me personally, I think the classification is okay. It's achieved quite high classification accuracy. But what is more important is, does it help us to understand the tumor biology better? So for instance, over here, when we visualize what is being learned, in terms of interpretability as a human trying to interpret the result, you see that CMS1, when we look at the morphological correlates over here, CMS1 is mucin rich, a lot of lymphocytic infiltration. And if you look at the RNA interpretation, right, it's also highlight that CMS1 is high in immune activity. So that's come out very well. CMS2, no one knows how it looks like before. And we show here that, hey, it's mostly correlated with cribiform growth patterns. Similarly, CMS3, no one knows how it looks like. And we show that it's mostly sort of branching glands over here. CMS4, of course, if you look at the RNA sort of interpretation over there, you would see that it's like the tumor infiltration, stromal growth pattern, and so on. And again, over here, we capture that very well in the in the patterns that strongly correlate with CMS4. So yeah, so that's to me, that's quite interesting. Any question before we go to the next part?
No, I think we're okay at the moment. So yeah, please carry on. Thank you.
Thank you. So talking about the unclassified samples previously, we didn't use these samples for training. But since we already have a classifier trained anyway, why not apply to this unclassified? So the reason for unclassified mostly in our data, TCGA and so on, is because of the poor materials. So it's not good enough it's not good enough for RNA or DNA read. So that's the problem. And we show over here that if we apply the image-based classifier on those unclassified samples, you would have a higher confidence as compared to the RNA-based prediction. Right, so what is showing over here is the distribution of the background molecular and genetic profiles. So it's because we don't have the CMS label for unclassified samples. So once you apply the image-based classifier on classified samples, the only way that we can check the possibility of classification is to look at the background of, say, if you classify sample as CMS1, you classify unclassified sample as CMS1, then you want to check the genetic or molecular distribution of those unclassified with the known molecular or genetic profile of the data that you already know in the class as CMS1. So the known one is in black, and the gray one is the unclassified that we have reclassified. You can see that mostly they match really, really well over there, except this one point from Grumpy, and I think it's CMS1 and in APC. But yeah, mostly, I mean, yeah, 99% they match really well. So that's quite encouraging for us. Any question, by the way? I think I might go a bit too fast over here.
So I suppose a naive question from me. So obviously you've said there's the 4 classifications and the 5th class is unclassified. So is that because what is the reason that they're unclassified? Is it, and it's because obviously you're now assigning them to one of those 4 classes. Are they unclassified because, yeah, yes. What's the reasoning? Sorry.
So it's molecularly unclassified. It's basically RNA classifier could not classify the sample, and the underlying reason is because it might be that the materials is not good enough to do the. So I mean, there may not be enough read.
Okay, so it's not like there's potentially a 5th class that's. It's not that way then.
Yeah, I mean it's possible. I cannot comment on that because I don't know either. That could be. I mean, CMS is one instance, but then you have like CRIS classification, which have 5 classes. But of course, the pathway involved, the biological interpretation would be quite different from CMS. Yeah.
Thank you.
Okay. So
Can I have a question, please?
Yes, of course, yes.
Okay. Can you go back 2 slides?
2 slides, yes.
Yeah, another one.
Yes.
Yeah. I'm not sure why it's being incorrectly, but anyway. Okay. About the morphology of the pattern, how prevalent are they? Because from what I understand, I may be wrong, but you classify CF3, CF4 at the entire slide level. Yeah, and then the carb-like pattern shown up, and then across multiple slides, and for example, across like a group of CF3 label from the route truth, what is the proportion of these patterns shown up on the entire slide across all the patients? Because it can, I'm just kind of like considering that because the network can sometimes hallucinate the prediction, and the pattern might actually have like spurious correlation. And another thing would kind of restricted to that is because you are patches-based classifier, right? You classify on based on the patches also, and it's another layer of like restricting the context. Your patches at the same site at when you perform at the same area that you perform the sequencing, the gene sequencing to get the route truth.
Yeah, so I'll answer I think the second question first, and then the first question, I think it's best answered in the next session of heterogeneity. So the second question where you ask, how do we know again, I might understand this incorrectly, so please do correct me, but if I understood correctly, is that you are asking, how do you know whether the ties that are predicted as, say, CMS4 is actually CMS4? And
yeah, that's not another way to, a more simplified way to say it.
Yeah, and as far as the data allow us to answer, we don't.
Okay, fair enough. Okay.
So the pattern shown over here are just like based on the prediction score that are highest correlated to CMS4, basically, or CMS3 and so on. So I think this is the part that perhaps you mentioned earlier about well, how like what is the percentage of ties being predicted with different classes? And going back to what other works has already published, saying that colorectal cancer is quite heterogeneous. We know recently from a French group, they work just published this year, actually, that CMS is quite heterogeneous. So there would be samples that have very heterogeneous CMS class, like what I show here on the top left, where this sample is entirely predicted as CMS2 in blue, while on the other one, you see that they are the tumor is quite heterogeneous in terms of CMS prediction. And from the work that I mentioned that's just published recently, when they look at, say, CMS2 class, separate the group that is homogeneous from heterogeneous group, and then look at the survival outcome, you would see that the heterogeneous group actually perform much poorer than the homogeneous group. So at the beginning, where I already showed that CMS4 has worse prognosis. If you actually separate CMS4 into homogeneous and heterogeneous, you would see this clear separation between the outcome. Similarly, in other CMS group. So this is quite, I think, quite interesting to look into in more detail. And in a way over here, because if you look at RNA or DNA data, it's a lump analysis. You scrap everything together, like you scrap all the material, you get one single readout. So the heterogeneity, the spatial heterogeneity is totally destroyed. Why the benefit of image-based, I mean, not only CMS over here, I mean, other image technique would have this benefit of maintaining the spatial heterogeneity, and I think this is quite important. This should be highlighted.
One question. This is the. I think it may not be available in your paper, but what is your perspective and your feeling with respect to the morphology heterogeneity of the CMS4 and CMS3, for example? Because even when we talk about the sequencing, you basically smash it and destroy all the entire variation of the tissue sample. But if they have high amount of heterogeneity, then we can hypothesize that on when you perform mass splicing for sequencing, it can likely show up. And if that's the case, then morphology within your, for example, within your the same picture A, whole soil image A, then the morphology of maybe the pinkish and the purple and the green one may have a mass distribution of morphology of tissue component are different. So what are you feeling about that? Because you may look at more images compared to me with respect to CMS3 and CMS4. So is there any like intuition or guess about what kind of like mutation manifests on which kind of like heterogeneity there? Aside from what coming up from the prediction, of course, because you already saw out the mucin already.
Sorry, I totally lost.
Yeah, okay. So, basically, for example, in the same slide, okay, we have a look at the one slide that contain like purple and yellows and green and different, and I mean basically different type like highlight there, right? Did you manage to check the tissue morphology using some specific metric that not based on the neural network? For example, nuclei typing, distribution, and for example, clan formation, etc., and so on. Even if not done quantitatively, because you already look at the image more than me with respect to CMS3 and CMS4. So what is your intuition with respect to the heterogeneity of the morphology of the tissue sample?
Yeah, okay, I see. I think we didn't look at that in detail except for clustering analysis. I don't think we have looked at the heterogeneity in terms of the clustering labels over there. So what we was interested in was how do you, I mean, it's good to look at the morphology, but how do you know that this is plausible? And the only ground truth that we have is basically to look back into the gene expression data and look at the heterogeneity over there, basically. Sorry, I didn't really answer your question well. I think
yeah, I mean, it's more about speculation about the rocklet rather than answering the rocklet. But I'm quite happy with both the outcomes. Yes.
Okay.
Yeah, thank you.
Yeah, so didn't do that, the short answer, but we did compare the heterogeneity between the image base and the RNA readout. And as shown in this subfigure B in the bars with different colors over there below that. If you look at the CMS, that's RNA-based classification, and the image-based CMS, and the bar represent the proportion of tiles being classified as different CMS class. Over there, you can see that mostly they visually correlate very well. We also do some statistic to show that this correlation is not due to chance either. So that's, again, I mean, that doesn't really provide any definite answer that the image-based actually capture the biological heterogeneity. But it's, I think, yeah, it doesn't rule out either that it doesn't. So with the result that we have over here, it's quite encouraging, and it shows that there is a possibility that we can capture that biological heterogeneity through the morphology. Yeah.
So we've got another question in the chat from Sarah, which sort of leads on from what you just talked about. So it's, were those heterogeneous samples correctly predicted? And maybe these heterogeneous samples were predicted as another class for a good reason and correlated with a different outcome, as the groups you mentioned.
So heterogeneity samples, are they, sorry, if you could remember.
Were the heterogeneous samples generally predicted correctly?
So the heterogeneity samples, of course, if it's like super heterogeneous, highly likely it would be unclassified. But those that are having like high level of heterogeneity, yet there is a dominant signature, generally it's I think it's generally performed well, as you can see from the AUC. But I think it's a good question. I need to look. It's been quite some time. Sorry. Okay. Sorry for missing. I think due to the voice through the mics, it's a bit difficult to capture the word. Yes. So the next step, I think it's the last one now, where you want to see whether the image-based classification actually still maintain the power of prognostic power of the RNA base or not. And over here on the left, there is the survival curve from the RNA data. And if you compare to the right, that's the image base. So the curve is per patient, not per patient, but at the patient level, not at the image level. So you can see that the curve correlate, the curve produced by the image base and the RNA base correlate very well over there. So we are quite happy with this. Shown only focus cohort, but if you want to see the result for other cohort, please refer to the publication. Yeah, and that's pretty much it. So if there is any question,
yes. Can you please go back to the previous slide?
Yes.
Yes. So here, this might be naive, but as I can see here, so the green curve and the pink curve, which are CMS3 and CMS4, they are pretty correlated. So this means that they cannot be stratified well based on these features. Am I right? Isn't it that in clinics these two groups are actually easy to be stratified?
CMS2 and CMS3 as compared to CMS1 and CMS4, I would say that CMS1 and CMS3 are quite
So, so what I'm saying, it's obvious that your features are doing a good job, or your features are doing a good job stratifying between CMS2 and CMS3, which were impossible to do before in clinic, which is really nice, right? But we still have some confusion in CMS3 and CMS4 for prognostication. Do you see what I'm saying?
So yeah, so it's not well known whether CMS2 and 3 are easy or differentiate clinically, because CMS is something that has not been applied in the clinic, right? From the RNA data, when we look at the evolution, they can go into a different path. So it's not like CMS 1 and 4 separate out before, and then you have CMS 2 and 3 in the evolutionary of the cancer. Is actually more convolved than that, because like CMS1 can be CMS1. No, I think CMS2, and then it can separate into CMS1 and 4, and CMS3 again. It can separate into CMS1 and 4. So there is a paper I can refer to that to you later on. So it's not clear to me whether CMS2 and 3 are clearly separated from the rest, because they are actually being part of the evolution of the disease.
All right.
Yeah.
Okay.
Brilliant. Thank you very much. Have we got any more questions from the people online at all? Quick look at the chat. So if not, I think that we can call it a day. But thank you so much. It's been a really interesting talk.
Hey.
You got?
Yeah, more
Oh, you've got. I thought them.
conclusion is fine.
Oh, my apologies. Sorry, Korsuk.
Yeah.
Did you have one? Did you have a couple more slides?
Oh no, so I think that's fine. So the conclusion is very much that the work has established that there is a correlation between histology and the CMS label, basically, and in terms of the implications, low resource settings, that can be done, I think, not the most priority. I think what we have done so far is to apply this to explore like all cohort or large scale clinical trials, and that's quite interesting. There would be works coming out, and the thing that would go forward in the next year is to apply CMS with collaboration with the Oxford Cancer Center over here, with the Oxford University Hospital, basically. So that's pretty much for this work. It's been quite some time. And we'll see the result in the next few years, I think. Yeah.
Oh, brilliant! That's been a really it's been a really good talk. So thank you very much for giving it. So we wish you the best of luck for the future for your work, and thank you also for everyone that's joined the meeting online. Hope you've all found it as interesting and useful as we have. And our next talk will be on Monday, the 16th of January at the same time. We'll be joined by Dr. Andre Hermeyer from Fraunhofer in Germany. So hopefully see some of you there. And if not, and I hope everyone has a nice and relaxing Christmas break. And again, thank you very much, Korsuk.
Bye. Thank you.
Thank you, Korsuk.
Thank you. Bye.